Welcome to this discussion forum. My name is Nick Reynolds. I'm Professor of Dermatology in Newcastle, and I've got the privilege of chairing this forum today, which is being run by the IEC, and we'd like to thank the IEC for their support in enabling this to go forward. You will all hopefully have seen the very good talks that have been pre-recorded. And so I'm going to ask now the speakers individually to introduce themselves. And then after that, we'll kind of have a wide-ranging discussion. There's no agenda, so it's completely open, and no one knows what's coming. Right, Bethany, over to you.
Hello, I'm Bethany Perez-White. I'm an assistant professor of dermatology at Northwestern University, and I'm co-director of the Skin Tissue Engineering and Morphology Core there at Northwestern.
Thank you very much. Killian?
Yeah, hello. Thank you, Nick. My name is Killian Eyrich. I'm professor for dermatology at the Karolinska Institute in Stockholm, Sweden, and also Heisenberg professor at the Technical University in Munich.
Thank you, Killian. And Reiko?
Yes, hi, my name is Reiko Tanaka. I'm a reader in computational systems biology and medicine in Imperial College London.
Okay, and I'd also like to introduce virtually, as it were, Jacob Thiessen, who may join us actually during this recording, and Sarah Brown, who's got other commitments today and can't join, but you'll also be seeing their talks. So Sarah's talk is on the genetic human models of atopic dermatitis. And I think that's a good starting point for us as a discussion panel, because that clearly underpins many of the changes that we see both clinically and histologically, as well as our research activities. And Sarah, as you will undoubtedly be aware, is an expert in particularly the genetics concerning the barrier function of skin and the importance of that in eczema pathogenesis. And I was struck by one of her comments during a talk, which indicates that only 20% of the heritability of atopic eczema is currently understood. And I was a little surprised about that, given that we know that certainly for our hospital-based patients, about 50% of them will have filaggrin mutation. So maybe we could start the panel discussion in no particular order, and people can just comment about what they feel about the barrier elements in the genetics and the overall contribution that we're seeing coming out in research. Killian, maybe you could start because it's an area of your interest.
Absolutely, it is. I think it's an area of the interest of all of us, but I think it's absolutely fascinating what you said, Nick. And I have two comments on that. The first one is that we have increasing evidence that barrier damage alone does not really induce inflammation. So filaggrin mutations, this we did know since a long time. They are, of course, causative in ichthyosis, but they are also happening and occurring in the normal population in quite a high number of percentages, actually. So this we did know, but more and more models now in the research, and these are, of course, murine models, but also human models, they illustrate that barrier damage alone, and that does not only refer to filaggrin, but to all kinds of barrier damage, does not necessarily induce an immune inflammation. So these two are in a way related, but they are independent. Then we do, of course, know that type 2 inflammation downregulates filaggrin. So type 2 inflammation has an impact on barrier, and probably also a little bit the other way around, but we need more to induce the full phenotype of atopic dermatitis. So that is one thing. The other thing is we know, of course, a lot of genes now which have a very low odds ratio other than filaggrin, that relate to atopic dermatitis. And the fact that all of them together account for only about 20% of the disease is highly interesting, I think. And it shows us that it's not genetics alone, but it's always a several hit model, I think. So it will be environmental influences, it might be epigenetics, it might be other factors that have to come in addition to genetics. And I personally think that very few patients have a more or less pure genetic phenotype. these are more maybe the ones which have monogenetic diseases like Netherton or men that come with a phenotype of eczema, but are not the typical atopic dermatitis patients.
No, I completely agree. And I think also the interaction of the environment, not only the kind of hard environment, if you like, but also the microbiome is so complex in eczema that I think that probably is a factor about A, the complete variability that we see in clinical presentation, and B, the relative progress that we're making compared to some other disorders that are perhaps where the environment isn't so influential. Because and I was struck as I heard all your talks that you were all fascinated by that interaction, but actually trying to model it is incredibly hard, isn't it? So to control for the humidity of an environment, the environmental exposure in terms of pollution, house dust mite, microbiome, and each one you have to take in turn if you're doing it as a reductionist model, or if you're trying to integrate it through Ryko's computational modeling. There's an awful lot to take into account, and we're not terribly good yet at measuring these elements in our longitudinal studies. And so I think that has been a hamper to the field really in making progress. Bethany or Ryko, do you have anything you'd like to say at this point?
Yes, so I completely agree that I like this more hits. It takes more than one hit to cause the disease phenotype. And so in my experience, experimentally, knockdown of filaggrin sometimes and sometimes doesn't cause an effect in the epidermis. So in 3D cultures using lentiviral knockdown, where you have a persistent knockdown, depending on the donor cell line. So we work with primary keratinocytes, and each time we run an experiment, it's with a new donor cell, new donor. So it's very variable. It's something that we have to deal with, but I believe it increases the rigor. But what we do find is that when you come in with a second hit, in addition to that filaggrin mutation, whether it's adding interleukin 4 and 13 to the culture, or coming on top with fungal extracts or house dust mite extract, then you can really see a ramp up of the phenotype. You see, spongiosis in the epidermal compartment of the 3D cultures. You can measure loss of barrier structure and function when it comes to both lipid and tight junction barriers. So yes, it's so complex in the underlying etiology of the patient, but also what's coming as an external insult. And so all of these produce a spectrum of phenotype that can be difficult to model, but Ryko's work is trying to attempt this. And so I think that that's something that we need to kind of tie all together and feed in even maybe some of the in vitro work into some of these larger computational models.
So Ryko, would you like to come in now and discuss then how, not in detail, but the kind of framework about the number of elements that you're able to include in your modeling, and how we can deal with the environmental aspects?
Yeah, so our computation models, for example, consider environmental factors, immune regulation, and barrier integrity. So these are the three main elements. And when we say environmental factors, it's quite conceptual. And there are lots of other things, the microbiome or the air pollution and all of these things. For the moment, we consider it as a one single entity. But when we have more data, then we can consider different types of the environmental factors. And similarly, for the moment, for the immune reactions, we considered adaptive immunity and the innate immunity as well. But obviously, there are more immune cells that might be playing a big role in atopic dermatitis. So it's like a hand in hand in terms of computation models and data accumulation and the accumulation of the knowledge. so it's quite important to work together so that we can get the informative data, hopefully the longitudinal for different time course and different time range, and that will help us to understand how different aspects of the disease happens.
Great. So may I move on to Bethany's talk in particular now? And I was very intrigued by the experiments where you found that the timing was very crucial for what happened when you added IL-4 and 13 to your 3D models. And I wondered if you could perhaps hypothesize why that may be, and you find this very distinctive difference. You may want to just briefly describe in case people haven't seen that particular element or don't recall it well.
Yes, so exposure to interleukin-4 and interleukin-13 in combination produces an atopic dermatitis-like phenotype in the 3D cultures. But depending on the time point that you harvest the sample, you can see different effects in the epidermal compartment. So at day 6, which is about halfway through the maturity of these 3D cultures, you see a loss of cornification and some spongiosis, whereas at the end of the 3D cultures, when they're mature by day 12, you see a very thickened stratum corneum, and there's not the flattened corneocytes that you would expect. And so I think this comes down to a balance between tissue homeostasis. So in those early time points, the epidermal compartment is still trying to make itself. So these 3D cultures arise from monolayers of primary epidermal keratinocytes. So it's the environment change when you put these at the air-liquid interface that causes stratification. So depending then on that time point is when you see the differences. But I like to take multiple time points because our skin is not always in a perfect homeostasis, and especially in an eczema patient that's constantly scratching, making wounds, you're going to have a lot of different states of the epidermis. So perhaps at the earlier time points, you're modeling a more of a hyperproliferative or a hyperactive epidermal compartment, whereas at the later time points, the keratinocytes have reached a homeostasis and differentiation. So you kind of need to take both separately to understand those different stages.
Great, and a related question then relates to your addition of your Th1 polarized lymphocytes. And I wondered if you saw a similar time issue with those.
So we only looked at later time points for that, and those were actually exposed for just about 72 hours. So the T cells were added to the media when the cultures were mature. So by day 9 or so, you have full stratification, you have full granular layer and cornification. So then you have a nice window about 72 to 96 hours to then insult that 3D culture to try to get a phenotype. So that's where those were.
And just technically, so you're polarizing them in solution, I presume, yes. And then you're adding, and how do you actually add them physically to the medium, or do you actually insert them into the dermis?
So as of yet, the polarized T cells we've only put into the media. So they're releasing factors that then, are going through the dermal collagen compartment and are impacting keratinocytes. So next we're looking to move those polarized keratinocytes into a collagen compartment through some sort of collagen stacking or culture stacking and that kind of thing. So we're really trying to figure out how to get those T cells into the dermal compartment where we think that they'll have a more profound impact on the epidermis.
May I add a question?
Of course.
Bethany, I'm also quite intrigued about these findings about the time course. And with that respect, I would like to ask you, what do you think about this we called it earlier a switch in atopic dermatitis, right? Where we said that type 2 inflammation is in the beginning, and then there was this initial postulate that there might be type 1 or type 17, Th22, whatever, at later stages. We then revised it a little bit and said it's not a switch. There is always Th2 dominance, but nevertheless, in later stages, you have other kind of immune cells infiltrating the skin, and they do the job which accounts for acidosis and in this chronic type of eczema picture. But from your data, would you guess that it might be the type 2 inflammation that does the job and it's just the kinetics? Or would you still say probably it's other immune cells than the type 2 cells that do it?
I think that it's really the type 2 cells, but I want to bring in the keratinocytes as some sort of immune reactive cells. So I think that over time, the keratinocytes are changing their phenotype and protein and gene expression, and is causing a negative loop. Like it's causing a bigger impact. And so, without the complexity of adding the immune cells, and that would be an amazing experiment, is to expose different types of T cells over time to the 3D cultures. But I really think that a lot of that has to do with keratinocyte effects to the IL-4, IL-13, and then that loop that keeps happening between crosstalk between immune cells and keratinocytes.
Okay, great. So can I then please move on to Killian's talk, whose focus was really the atopic patch test and its applicability to clinical practice as well as research. And from my understanding of your talk, you emphasize the limited applicability that it's really made, despite enormous promise in clinical practice, in part because of the issues around standardization. And I wondered if you could just briefly describe to us what you feel is the optimal standardized form that you would recommend in the research context, where I think there's many experiments that we could all think that would be still great to do using that and different allergens in different contexts.
Yeah, that's a highly complicated question, of course. I don't know how you feel about it, right? But what is clear is that the atopy patch test has a clinical impact also, I think. It's just so super hard to standardize. If you put it on the back of a patient, a totally different reaction might come out than if you put it on the underarm, for example. So this standardization is, of course, a big problem for research questions also. So my first, let's say, danger warning would be ask the right question if you want to put the atopy patch test in place here. And if it is related to early inflammation, inflammatory infiltrate, to antigen specificity, then it might be absolutely the right test. You need to have a kind of standardized way how to applicate the antigen, the allergen. And then, of course, there are questions such as, do you interfere with the microbiome? What happens if you do occlusive therapies? What kind of patient do we have? There are other reports, right, that Vaseline itself is very good for atopic dermatitis, occlusive for 48 hours. So you will have quite an interaction with a lot of factors. So that means first define the right question, then standardize the setting as much as possible. And the third part would be know as much about the patient as possible. So in what kind of patient state is the patient? Does he have a flare-up right now? Is it a patient that has a filaggrin mutation or a strong barrier defect? Or is it a patient in the pollen season, for example, that gets an aggravation, and we do the pollen patch test here in a kind of T-cell exhaustion state? So these three questions, of course, need to be answered. And I think that the most important one of that is ask the right question. What do you want to know? How much does an antigen, for example, contribute to an early reaction? How does an antigen contribute to the more chronic state, as we have just discussed? Bethany has just illustrated to us. Probably you would never ask something about microbiome, because the setting is that you have occlusive patch testing for 48 hours. So you would definitely change the microbiome itself, right, in that kind of setting. So it is quite complicated still, but I still think that the atopy patch test has given us breakthroughs, especially in terms of the immune response pattern, and also in terms of some little more chronic pictures like late stage infiltration of immune cells. And these can be informative, for example, for experiments that Bethany could do in vitro, but of course also for Raiko that could add this in her algorithms.
Great. So Bethany or Raiko, do you have any points you'd like to discuss with Kilian?
I was just wondering if anyone has used the atopy patch testing in normal, participants, normal subjects, to, ask the question. So, the differences between the back and the arm, well, there's probably definitely microbiome differences there. So is there any insight from normal subjects?
Yeah, very early studies have done that, and they report a very good specificity for AD patients. In other words, non-AD patients usually do not develop a kind of reaction. There are also some exceptions. For example, there are house dust mite preparations, which are very irritative. And then depending on the localization, depending on the microbiome, as we say, depending on the thickness of the epidermis, and on the general state of the skin, you might get irritant reactions as well. But the true allergen-specific positive atopy patches have a very high specificity towards AD patients.
Mm-hmm. Yeah.
And can I just explore a bit further with you the issue of the filaggrin barrier dysfunction with atopy patch testing, and whether that has been explored systematically, either at an individual allergen level or in terms of the level of reactions that you may get for a given allergen stimulus?
Yeah, of course, that's a very interesting question, and it has been tried several times. The problem is that the standardization, the heterogeneity of the patients is so high that probably you cannot get a clear-cut result here. So there's I couldn't tell you now a study which clearly shows that filaggrin mutations have a higher degree, either severity or incidence of positive atopy patch tests, although we would assume that this might be the case. The heterogeneity in the human system, in this very complex system, is obviously too high to get this answer.
Okay, so that really brings us on to systems biology then, and I think we've already alluded to the fact that we do need to take a systems approach to eczema in order to try and integrate all the different signals that are coming in from the environment with the genetics and the inflammation. And I was struck by Raiko's talk in terms of the levels of models that are now available to us. And I wonder if we could start with a general question about whether you think that the modeling that's gaining great prominence in COVID-19 has actually reinforced for the field that we should be taking modeling more seriously, both at an individual and population level.
Yeah, I think that was a very good example, and lots of different models were developed during a very short time period, and then the data was available, data was shared, and that was probably a big advantage of this era. And then people appreciated the modeling, and yeah, the prediction was reasonable and understandable. So I think lots of people begin to realize the power of the mathematical modeling and how the different data could be combined to make the actual prediction and actual recommendation.
So Bethany or Glenn, do you have any comments about that and modeling more generally in eczema?
For me, the heterogeneity is here the really important point, right? If I understand correctly, Raiko, the models are really pretty far advanced, and I was impressed to see how far they are advanced, in fact. But in the end, it all comes to the metadata, right, that you present in terms of clinical phenotyping and in terms of where the biopsy has been taken, under what conditions, for example, to clean up the system. Would you say if you just have enough data, and that relates a little bit to the COVID, right? If you just have, let's say, 1 million sample data of lesional skin of AD, all this doesn't matter? So could it be anywhere, any patient, any genetic phenotype, but you can still get signatures out with your models? Or would you say we as dermatologists have to be more precise and give more metadata, more phenotyping to the material and the data that we are sending in on a molecular level?
It really depends on the type of the questions that you want to ask with the models. And so it's kind of the chicken and egg. The type of the questions that you want to ask will determine the model, and also determines what kind of data that we need. And also, most likely in the clinical setting, what kind of data are easily available with a certain precision. So all of these are combined, and then that would determine, we might want to answer this question, but it's probably better to aim for this kind of the questions so that we can get enough data and we can get some kind of the answers. And in terms of the personalized models, so we developed the mechanistic model, which is quite like a metadata type of things, but we also use the patient-dependent actual severity score data, and the model can predict the individualized severity prediction. So depending on the types of data, we can make the model personalized, or we can focus more on the metadata or meta mechanism type of things. And with the strong increase of machine learning tools, this kind of personalized models development become more easily possible.
So I wonder if you could expand on that a bit more, Raiko, in terms of, because I think the field's very interested in the concept of personalized modeling. If you could present whatever data was required for a patient, and you could either optimize the drugs that you would use, or the way that you use them, or how often they're applied, or the frequency of dosing, that would be an enormous help to us in the clinic. Yeah. So in order to develop a robust individual model, what kind of data are you requiring? It goes back to Killian's point, is it in terms of what is driving the response, the variation in response?
So it again depends on what we want to achieve by the treatment. If the aim of the treatment is to decrease the severity scores, then we should definitely measure the severity scores. The aim of the treatment is to try to decrease certain kind of the cytokines, IL-4, IL-13, then we should measure these cytokine levels more carefully. More carefully means at more time points. And then if we can combine all of these together, like the cytokine levels and the molecular levels and the severity scores level, then we may be able to make the model which can combine all of these different kinds of data, and maybe transcriptome data as well.
Yes, but I would suggest that because one of the ultimate aims really is to be able to, at a certain point in time, which in the study is called baseline, but in the clinic can be any phase in a patient journey, to take some biological sample, assess their clinical phenotype severity, and then end up in a way of endotyping, if you like, that patient, and then being able to recommend a particular bespoke treatment for that individual. And I would suggest that the field in general, although it can collect now, RNA-seq, single and bulk data, cytokines data from blood, what generally is lacking in when you come and try and synthesize the whole, it is the clinical metadata. There's an availability to the field. So you may be able to get the RNA-seq from geo, but if you try and combine it at an individual level with the change in their EASI scores over time, that's much harder to synthesize. And I think you're quite right that you need all of it in order to be able to reliably predict at an individual level what's going to happen to them.
Yeah, and currently the severe lack of the time course data is a problem. The transcriptome data, RNA-seq data can be obtained at baseline, and that's it. But it's so noisy to predict any kind of the development, and AD symptoms develop so quickly and so randomly over the time. So one single time point measurement is not very predictive in terms of the treatment prediction and treatment effects prediction for the moment.
So great. I think I really enjoyed our discussion. Thank you very much for all your contributions. Does anyone have any final points they've not said yet?
In the end, it comes to, we have to combine all the methods we can get because of the high heterogeneity of the disease pathogenesis and phenotype, right? I also enjoyed it very much. Thanks, Nick, for moderating this.
Thank you also, Bethany and Raiko. And again, thanks to the IEC for organizing it.